In 2011, researchers at the Enhanced Animal Biosafety Level 3 facility at the Erasmus Medical Center in Rotterdam witnessed the emergence of a new and possibly unique variety of flu virus. Concerned with the emergence of a new flu pandemic, they were studying how viruses change to pass between two different species.
I will start to say that I'm in a more or less easy position. Talking from a sort of ivory tower, we use all the technology that we are talking about now only for science. And working in the Netherlands Cancer Institute, you use it also for doing good science to solve problems in human healthcare. But I experienced in between, I worked there till 1990, and then I moved to a company called Farming, at that time GenFarm, and we started working in a commercial setting. And then we felt how the public was annoyed by our enterprise to make commercially attractive biotechnology species, and that raised a lot of controversy. And then we had to defend ourselves on the background of a commercial setting that is completely different. The thing is now, GenFarm generated hormone de Boel, and that was for well, all kinds of things, for food production, but also for pharmaceuticals. But the same company, at that time, it felt a lot of controversy and was in the public arena. It was very much opposed to. But the same company started also at the same time working at a disease called Pompe disease. And everybody knows now in Holland that the pharmaceuticals that were generated by farming to treat patients having Pompe disease, that worked well, but now we don't have the capacity anymore, the financial capacity anymore to pay that pharmaceutical. And now everybody is again very much against the decision that we shouldn't pay for that pharmaceutical. So in between 20 years, the public opinion changed from radical against to a more or less radical in favor, not knowing what they were discussing. It was the same company. So that was about that company. And after that I left there, I joined the Netherlands Cancer Institute again, and I feel the atmosphere of the ivory tower again, doing only science, doing only fundamental science, so to say, to know more about the generation of cancer, metastasis, and so on and so on. And we use these technologies that I will talk about now, just to improve our models, to mouse models, to study these diseases. So, as I said, we work with mice, and when we talk about GMOs in animals, Richard has a lot talked about GMOs in plants. I will talk only about animals because my feeling is that they are more sensitive to the public opinion than plants. I will talk first about mice. There it all started. I will talk about transgenics and knockouts. And all the technology that is developed in mice is now being used also in other species. And fortunately, the other species are much more complicated to work with, and it takes a lot more time to be efficient. So we have a lot more time to think about it, what we are doing. So that's fortunate about these higher, larger animals. Now, knockouts and transgenics. What is a knockout? A knockout is a species that misses a gene or multiple genes. So it has gene loss, and you can have it conditionally and you can have it constitutive. Constitutive, all the cells miss that gene, and the gene is deleted already in the germline. A conditional knockout is a knockout mouse is a mouse which is at will knockout in a certain tissue, the brain, the liver, the blood, wherever, at a moment where you want to have it deleted there. That's a complicated technology, but it's possible. Transgenics, in our nomenclature, is a species, is a creature which has additional genetic information, and that can be multiple genes, that can be only one gene, combination, and so on. I have to have my glasses. So it can be a foreign gene, which is foreign to the species which you are inserting it. It has multiple copies, and you can have it expressed in other tissues than originally. You can take, for instance, hemoglobin, which is only present in blood, but you can also express it in the liver or in the brain. Scientifically, it doesn't make sense, but okay, you can do it. So, what kinds of questions do we want to answer making transgenic animals? First of all, you can just basically study gene regulation. So a gene consists of a structural, it's a linear information. It's just a linear molecule of DNA. And you have structural information in the DNA which encodes the protein itself, but you have also regulational information which controls when, how, and where it should be expressed. And this regulatory information is not very clear on the basis of the DNA sequence. So what people do is they just take a piece of DNA which encodes the gene, the protein, and they just put it into a mouse and study where it is expressed. Has it sufficient information surrounding this structural information to get it expressed properly, then you have the complete gene. If not, then you have to extend the DNA on both sides to get it expressed in a normal way. If you do that, then you can use these transgenes to study the biological consequences. And you can combine that with gene knockouts and with other mutations. First of all, and primarily, these mice are used to make models for human diseases, and that's what we do for cancer. And so you can, then mutation that you find in a certain disease, in our case tumor, you can use those mice to confirm that mutation is causally related to the onset of the tumor, or causally related to, for instance, the onset of diabetes, or any other disease. Subsequently, you can use these models to try to set up a therapeutic intervention in these models. That saves a lot of work doing these preventions, these therapeutic intervention studies in humans. So you can select your therapeutic intervention protocols first on the basis of what you do in mice, and then subsequently you can do that in humans. And that's what we also do in the Netherlands Cancer Institute. First we create the models for certain diseases, and subsequently we test them for therapeutic intervention. Now there are several methods to make genetically modified mice. Basically there are two methods. The first method is just straight zygote injection. That's the most simple one, and it was already done some 30 years ago. And you have a cloned piece of DNA which encodes your DNA, your gene of interest, and you just directly inject it in the pronucleus of a zygote. A zygote is a merge between the oocyte and sperm, and when the sperm enters the zygote, at a certain moment you see two nuclei, one of the oocyte and one of the sperm, and before real fertilization has taken place, they have to fuse. And before fusion, you can see the individual pronuclei, and it's very easy to inject a couple of microliters, picoliters I should say, into either of the two pronuclei. Well, after you have done that, and you inject basically a few hundreds of molecules of DNA into that pronucleus of a single gene, after the injection you implant these injected zygotes into a pseudopregnant female, and after 3 weeks, in mice at least, the pseudopregnant females will deliver the pups. And of course, then you don't know if your injection was successful or not. You have to test the success of this injection by taking a piece of the tail or some blood and see if your gene of interest is indeed in the genome of the mouse. That's basically the approach that is taken to make transgenic mice. So you get an addition of genetic information. The second method is based on the existence of ES cells. And what are ES cells? ES cells are cells that are capable of contribution to any tissue in the body. So they are totipotent. So if you would introduce ES cells into a developing embryo in the blastocyst stage, that's a stage before implantation, then these ES cells, they take part in the embryo proper. Also in the germline of the embryo that is being generated. Now if you have mutated these ES cells in vitro for a certain mutation, for instance a knockout or an addition or whatever, part of the cells of the developing fetus will also exist of cells that have that specific mutation that you have inserted already in the ES cells. That saves a lot of mice that are not successful in having these mutations, because you have already selected for the mutation in vitro. This is in contrast to the first method, where you have to wait and see if your manipulation was successful. Now, first, the injection. The first transgenic mouse was published 30 years ago. And that was a mouse that had the fusion gene between a promoter, liver-specific promoter, hooked up to the genetic information for encoding growth hormone. And as you can see, one of the two is transgenic, and I guess the right one is transgenic. It's much larger because there's a lot more growth hormone in his blood than the left one. That's his littermate, which was not transgenic. So that was the first successful transgenic mouse having a phenotype because of the insertion of additional information. Well, many other mice have been generated. For instance, this is the GFP mouse. GFP stands for green fluorescent protein. This is a mouse having a protein which is expressed all over the body. And as you can see, if you put it in a UV, under a UV light, then it glows up, and the left one doesn't. This is not a very sensitive mouse. It makes no sense to make such a mouse. But you can use these GFP transgenes to follow cells where they end up in the body. For instance, if a cancer cell would glow like this, you can follow, for instance, metastasis. When it has a mutation, when it has a tumor in the colon and it metastasizes to the lungs, for instance, you can see that already from the outside. So therefore, these transgenic GFP mice are being used. There are several other expression studies being done. This is a mouse which is expressing LAG-Z. LAG-Z is an enzyme that metabolizes a metabolite into a blue protein. And when you want to study, for instance, the promoter of a gene, where it is specific for, then you hook it up to the LAG-Z gene and you stain the embryos for LAG-Z, and then you see that this promoter was driving the expression in the back of this fetus. Now, basically, what you do with transgenics is the following. The upper part shows schematically the transgene. The first part is a promoter. The second, the blank part is the coding sequence, and on the right you have also some regulatory sequences. For instance, the poly-A site is necessary for a signal to stop transcription. Now, this piece of DNA, you can multiply it to enormous numbers, and you have just a solution then with only this molecule. This molecule then is put into a needle, injected into the zygote, into the pronuclei, either the male or the female. These zygotes, they come from superovulated females and they are mated to males. On the morning after mating, these zygotes are isolated from these females, and you can just see in the microscope if they are fertilized or not. When they are fertilized, they have 2 pronuclei. If not, they don't have any pronuclei. Now, you inject these embryos, and 1 day later, they have divided into 2 cell stage. And these 2 cell stage embryos, they are implanted into the oviduct. And the oviduct is an oviduct of a pseudo-pregnant female, which has nothing to do with the male, which has been used to get these oocytes. This pseudo-pregnant female has been mated by a sterile male. So she thinks, I'm pregnant. She's not, but we will make her pregnant by the implantation. Now, after 3 weeks, pups are born, and you do tail biopsies. And these tail biopsies are used to isolate DNA and to get to do the analysis for transgenesis or not. Now, after again 5 weeks, if they are transgenic, then these transgenic mice are fertile, and you can make a line of these mice, and then you can start your study. Now, something about some numbers. We inject 0.5 DPC, days post-coitum, does it mean? Super-ovulated or natural mated. Typically we inject on an afternoon 200 zygotes, and about 80 to 90% will survive the injections. Some will be destroyed because you have to. It's a micro-manipulation, you can destroy them. And then the DNA is usually injected in the male nucleus. And this is what shows you. Many people, of course, have already seen this. On the left you have a needle which is inactivating positioning the zygote, and on the right you have a needle which is inserting the male pronucleus. Now, after transfer and delivery of the pups, it appears that almost 30% of the transplanted embryos develop a term. So if you implant, let's say, 100 embryos, you get in between 25 and 30 pups born. And the success rate of transgenesis depends very much on the gene that you inject. 2 to 30% of the pups, live-born pups, is transgenic, have the additional information. Now, and then you start making lines of these individual transgenic mice and do the experiment that you want to do. Now, the other method is based on the existence of ES cells. Now, embryonic stem cells, as I said, they are totipotent. They contribute to all the tissues of a mouse, and therefore also to the germline of a mouse. And therefore, if the germline contains cells that are derived from mutated ES cells, they will transmit the mutation to their offspring. These ES cells are harvested from a developing embryo, and the schematic is shown here on the left. You have a day 1 embryo after fertilization, 2 cell stage, day 2, a morula, which is a clump of cells which you cannot really distinguish the cells in. But after 5 days, the embryo looks like a big ball, and in that ball there's a big vacuole, and you have a rim of cells here, which is called the inner cell mass. And the inner cell mass will ultimately lead to the embryo proper. And the ES cells are derived from the inner cell mass. You can extract the inner cell mass from such an embryo, put them in culture in specific conditions, and making a continuous cell line of it. And using different factors, you can maintain the ES cell in the ES cell stage, in the totipotent stage. But also, you can manipulate the ES cells. You can put DNA in, you can do selections, and so on and so on, still maintaining the totipotency of these cells. These cells look like this, Here you have these little islands of cells. These are the ES cells, and underneath you see also cells. These are fibroblasts, and they support the maintenance of the totipotency of the ES cells. Now this is very old-fashioned. Now we have culture conditions where we don't need the fibroblasts, and we have real nice only ES cells in our dish. Now these ES cells, they are exposed to DNA. We make holes in these cells. They still survive. And we do that by electroporation. They get a lot of DNA in there, and the nice thing about DNA is DNA likes other DNA. It will stick to each other. Incoming DNA will stick to each other, and they recombine with the endogenous DNA. And this recombination with endogenous DNA, that gives you the opportunity to insert DNA. And the insertion you can select for. And it's not very efficient, but because of the selection, it ultimately is efficient. You can end up only with cells that you really want to have. Now, this is what I mean by that the DNA likes each other. Here are two strands of DNA, and they are very much alike. And because they are very much alike, they will recombine with each other. Now, if, for instance, the red one has a sort of mutation or an additional information, and the blue doesn't, the blue is endogenous, then after recombination, part of the red will be inserted in the blue. And therefore, you can just insert new information in the endogenous genome. That's the principle of manipulating ES cells. And this is called homologous recombination. And, well, this looks a little bit more complicated, but the principle is the same. Here I show you a typical vector, as it is called, which enables us to select for the event that you want to have. I don't go into details about these vectors, but we have now vectors that you can really use for efficiently introducing small mutations, large mutations, whatever you need. And all these selection markers like NEO and TK are used for positive or either negative selection. To make a long story short, using these vectors, we can just introduce the mutation that we want. Now, typically, it is more or less the same as the zygote injections. After having done the mutations in ES cells, and after screening with the proper ES cells, at day 1 we inject the blastocysts with the ES cells. Then these blastocysts are also implanted in the pseudopregnant females a little bit later in their cycle than the zygotes. But then you get mice, of course, that consist of cells that are derived from the host blastocyst, but also from the incoming ES cells. And these mice are called chimeras. This is a typical injection. You have also shown that already. You have a film, and here's just it's about the same. About 15 cells are injected in the blastocyst. Now, after having given birth by the fosters, you get either black mice, which don't have taken up ES cells, so there the injection failed, or you get chimeric founder mice, and these mice consist of mice derived from the ES cells and from the endogenous black 6, the host blastocyst. The middle one is a very nice chimera, but the lower one is a little bit less chimeric. So the upper one, the middle one, has apparently the development of that mouse is based on development of tissues which were derived from the ES cells. But the lower one is much less successful, and breeding of these mice will show if also the germline has taken up cells that are derived from the ES cells. You know, two also very nice examples of chimeric mice, and you get these stripes. This looks like a lagervelder, but you get all kinds of patterns. Now, this is what we can do in mice. So far so good, and this is also guided by legislation, ethical evaluation. Also there are rules to maintain the mice in containment. There are large databases existing. All the mutated mouse lines have been registered, and I'll show you an example of a repository organization called EMMA, which is based in Rome, and there you can deposit your mutant lines for cryopreservation, but also for distribution. Here we have contacts with this organization, and they will have about, I think, 1,000 to 2,000 lines that were generated in Europe, and the labs, they just transfer these lines to this organization, and they will make stocks of them, either in liquid nitrogen or just as living mice. Another organization is this organization, the Jackson Lab. They have many more mice. I think they will have 50,000 of lines, mouse lines, and you can also order mice from there. And they have an order form which you can use to specifically order mice. Every mouse that you order there is basically for free, for fundamental research at least. You only have to pay for transportation. So if you want to have a mouse for certain purposes, you just go to the website and see which gene would I like to have mutated, and if it is there, you can just order it there. There are also other websites that you can visit. There is a website from UCOM. UCOM is an organization in England, and they have knocked out all the genes that exist in mice. And if you want to make a mouse, then you order just the mouse. If they haven't already derived the mouse, you just order the ES cells and make the mice yourself doing this microinjection. Now this is, of course, very efficient to order mice or ES cells, but is that what you, sometimes that is not what you really want. What you get there is a complete knockout generated in a specific way. But sometimes you want to have some mutation, a very specific mutation, which is conditional or just a point mutation, and that is something you have to make yourself. But having these technology available, you are able to do that. So for mice, it's very efficient to make the strain that you really want. Now, this is just an example how efficient this is. Just yesterday I got this email from the Jackson Lab, and they announced to me that if I want to have one of these strains, I should be quick, because now they have them as livestock available, but in a few weeks they will only have them as sperm or as frozen embryos, and then I have to make the revive them myself. So if I'm quick and I go over all these links, each link is representing a mouse line. And if I want to have them, I should be quick, then I can just get the live mice, which is much cheaper than ordering embryos. So this is how it is in fundamental research, and this is open, it's public, and it's very easy and accessible and also understandable. This is very transparent. When it comes to other species, it becomes more difficult. So mice have been used to start off with because mice are cheap, they have a very short regeneration time, they breed like crazy, it's very easy to get many mice, and also the embryology that is necessary to do all this was already available. Also, because of the embryology, stem cells were available. So because of the knowledge that was already there, and mice were chosen, and also for economic reasons, the mouse was chosen to develop the technology. Of course, there are other species which are much more interesting to many people in society than just mice. Mice are interesting for fundamental science. Other species like dogs, cows, and so on, they're also very interesting, but their technology is is lagging. And people, of course, like Rich already alluded to, have just gone through the selection procedure. And that's also intervening in normal evolution. But now we can do that much quicker. Selection takes forever. The cattle that we have at the moment, it took at least 1,000 years to have them producing that much milk that they can do now. At least 1,000 years. And now we can do that within 1 generation. So it speeds up the evolution enormously. And that is a worry because it speeds up, we cannot think about it, what we are doing. So that's a problem in itself. Although, of course, it has also many pros to speed up evolution, because for some questions we cannot wait. For food production we cannot wait forever. But for evaluating what the real consequences are for nature, it might take a little bit more time than just 1 generation. And we don't get the time for that. Now these are the species that are now being used for making transgenic individuals. In the middle, that's Herman, the bull. And for cattle, at the moment there are no ES cells available. Just to illustrate how we made Herman the bull, short story. In '88 we had the idea to make, that company had the idea to make a transgenic cattle. And how do you have to start somewhere, you have to start with an embryo. How to get so many embryos that you can inject. And the approach that we took was, we asked all the slaughterhouses in the neighborhood of Leiden, where Gen Farm was based. And there was Rotterdam, Leiden, Haarlem, Woerden for that matter. And we got all the ovaries from cattle that was slaughtered at a particular day in a bucket. We transported to our laboratory, and we aspirated all the follicles visible on an ovary. And an ovary of a cow is like this. Sometimes they're a little bit bigger, but you can easily see the follicles. The follicles that were more or less at the point of maturation, those were the follicles that we took. And they were that big. So we were sitting with a crew of 5-6 people, getting 1,000 ovaries each day in a week, and we were just aspirating these oocytes. And these oocytes, they were not mature yet. And it's funny to talk to these guys at the slaughter line, because they, what ovary? What is an ovary? Eierstok? Didn't know what we were looking for. And when you open up a cow which is hanging on these hooks down, and they open up the belly and everything comes out, the stomach and the intestines, and also the kidneys, but also the ovaries. So they had to look for the ovaries in this. There was this big, warm. Everything was warm, of course, and we got 2 ovaries for each cow. And each cow, at that point, we had BSA in Holland, was also registered with a barcode. So all the ovaries that we got were barcoded. But of course we put them in a bucket, and we didn't follow what happened to the individual oocytes. Now these oocytes, we had about 500 each day, and we matured them to fertilizable oocytes, and therefore we used a straw of famous bulls, and we fertilized them in vitro, because otherwise we couldn't get zygotes. So these these bulls, they were Sonny Boy and Trogar and all the famous names by that time. Sonny Boy, I think, generated the whole cattle in Holland. So that was a very popular one. And all these fertilized zygotes, they were injected a day later. And then we had, of course, to implant them in fosters. Well, a foster like mice, that's easy. But a foster in cattle is also a cow. So these cows should be tochter. in order to be a proper recipient. And tochtig, yeah, tochtig is a certain phase in the cycle of a cow. Yeah, yeah. So all these fertilized, all these injected zygotes, they were matured to blastocysts, and then we went up to Lelystad, where a big farm was and with a lot of cattle, and they organized the recipients, and in implantation in mice is easy, but in cattle, that's you go in like this, and then you have to find these oviducts, and to inject basically the injected embryos.
Did you do it?
No, I didn't do that. No, my arms were too short. So, and then 9 months later, all the animals were born, and we had to take biopsies to find out if they were transgenic. So that was in the old days. What they do now is they have a cell line of a cow, and what they do is they extract the nucleus of those cells, and they merge it with an oocyte, a nuclear transplantation. And if you have manipulated these cell lines, you already know if it is transgenic or knockout or whatever. And in contrast to what we did, we had to inject many zygotes and wait for 9.5 months to find out if it was transgenic or not. So that's far too expensive. What we do now is much more efficient, reduces the time to make all these mutations that we want. That's fine, but it reduces also the opportunity to think about it, what we are doing. So, yeah. Oh yeah, there's a lot of things that, oh yeah, okay. This is not so important, but this is only to mention that all these pets that are generated, they are not generated in the Western world, but they are generated in other countries. So we are very sensitive about making transgenic animals. We still are, but it is a lot more easy, but we are still sensitive. We discuss a lot about it. It has the goal why you do this should be a decent goal for production, healthcare, or whatever. But for using them as pets, that's not acceptable in Holland and also in the rest of the Western world. That shows that we are thinking differently about making transgenic animals than other societies. I'm not saying that they have a wrong opinion about it, but yeah, they are doing things differently. And if we then release all these mutated organisms in public, then we have to think about how other societies deal with that. And there should be some way of, there should be some consensus about it if we should do that. Okay. So, I think that the topics that we should discuss is containment, and containment is just to have full control over where all these species, all these animals or plants are going. We should just think about globalization. What is the opinion about other societies? Ownership, Rich has already talked about it, how companies are trying to protect themselves and to restrict the use of them by others. We have a much larger, bigger responsibility towards nature using all this technology, and we have not a lot of time to think about it because the pressure is very high. And also, doing all this, it is giving direction to evolution. The evolution so far was not directed. Of course, selection, what we did some decades ago, was also directed, but there was still a result that you cannot predict. You just select what you like and discard the rest. Now, you first define what you like and you just make it. And that's completely different. So the directional evolution is much more taken off than previously. Okay, that's what I want to say.
The sea monkeys displayed here were originally created by Harold von Braunhut and Dr. Anthony D'Agostino at the New York Ocean Science Laboratory in the early 1970s. The project created a hybrid brine shrimp variety that has an unusually long period of dormancy and can survive in an airtight packet for months or years at a time. The continuous financial success of sea monkeys starting in the 1970s stems from von Braunhut's strategy of marketing directly to children through an unprecedented number of advertisements in the back of comic books. In 1999, sea monkeys were marketed in Greece for the first time under the name Little Sea Souls. A member of the Greek parliament attempted to block the product on the grounds that it is unethical to sell a living organism as a toy. Concerns were also expressed that the brine shrimp may have been genetically modified. When the investigation was complete, Little Sea Souls were finally approved for sale in 2004. Harold von Braunhut died in 2003.